Good morning, everyone. Today topic is polymerase enzymes. Polymerase enzyme. So in this video lecture, we will cover polymerase enzyme and types of polymerase enzyme in prokaryotic cells and types of polymerase enzyme in eukaryotic cells. So generally, polymerase enzyme are two types. They are DNA polymerase enzyme, DNA polymerase enzyme. Second one is RNA polymerase enzyme. Now, this DNA polymerase enzyme helps for synthesis of DNA. They helps for synthesis of DNA. These RNA polymerase enzymes helps for synthesis of RNA. So here we have two DNA polymerase enzyme again divided into two types. There are two types. Yeah. Two types. They are DNA dependent DNA polymerase enzyme, DNA dependent DNA polymerase enzyme, and one more type, RNA dependent DNA polymerase. And here RNA polymerase enzyme also two types. They are DNA dependent RNA polymerase enzyme, DNA dependent RNA polymerase enzyme. Here second type, RNA dependent RNA polymerase enzyme. So we will discuss each type in detail. First, DNA dependent DNA polymerase. So this DNA dependent DNA polymerase generally found in both organisms like prokaryotic organism and eukaryotic organism. So, and in which DNA dependent DNA polymerase, they helps from DNA to DNA. So they helps in synthesis of DNA to DNA. It means this process called replication. So the process of producing two identical DNase replicas from the parental DNA the process called replication. So the replication process is completely depends on DNA dependent DNA polymerase enzyme. Now coming to RNA dependent RNA polymerase enzyme. Here I am writing. Second one, RNA dependent DNA polymerase. And these enzymes are mainly found in viruses. Found in viruses. Example like HIV virus and RSV virus. RSV means RAS sarcoma virus, hepatitis B virus, and very important, cauliflower mosaic virus. So in this virus, there is a presence of RNA dependent DNA polymerase enzyme. So in this RNA dependent DNA polymerase enzyme was first discovered by two scientists. They are Temin and Baltimore. Temin and Baltimore in 1970 in a virus called RAS sarcoma virus. So in which RNA dependent DNA polymerase enzyme, here from RNA there is a synthesis of DNA. So generally in replication process or in transcription translation process, generally from DNA to RNA synthesis will take place by transcription process, by transcription. But here from RNA there is a synthesis of DNA, means it is a reverse transcription process. Reverse transcription process. Hence this enzyme is called reverse transcriptase enzyme. So this RNA dependent DNA polymerase enzyme also called reverse transcriptase enzymes. Reverse transcriptase enzyme. So this is all about the DNA polymerase enzyme, in which DNA dependent DNA polymerase enzyme and RNA dependent DNA polymerase enzyme. Now we will move on RNA polymerase enzyme. Second type, RNA polymerase enzyme. So these enzymes mainly helps to synthesis of RNA. So here also we have two different types. One is DNA dependent RNA polymerase, DNA dependent RNA polymerase, and one more RNA dependent RNA polymerase enzyme. Okay. This DNA dependent RNA polymerase enzyme generally found in both organisms like prokaryotic and eukaryotic. Hence they are present in prokaryotic eukaryotic RNA polymerase. They are present in prokaryotic and eukaryotic RNA polymerase enzyme, and in which from DNA there is a synthesis of RNA. So this is what transcription process. So by transcription process, the DNA undergoes transcription to synthesis of RNA. So hence this DNA dependent RNA polymerase also called transcriptase enzyme. Also called transcriptase enzyme. So in presence of transcriptase, a DNA dependent RNA polymerase enzyme. So here there is a synthesis of RNA from the DNA. And one more example here we have. So that is from DNA there is a synthesis of RNA primers. There is a synthesis of RNA primers in presence of an enzyme called primase enzyme. Primase enzyme. So this enzyme is also comes under DNA dependent RNA polymerase enzymes. And these RNA primers helps in DNA replication. Helps in DNA replication. DNA replication. So this is all about the DNA dependent RNA polymerase enzyme. Now coming to RNA dependent RNA polymerase enzyme. So these enzymes generally found in RNA virus, RNA containing virus. RNA containing virus. Example, poly A polymerase enzyme. Poly A polymerase enzyme. So these enzymes helps in formation of RNA from the RNA only. So from in which RNA gives rise to RNA synthesis. So from parent RNA there is a formation of daughter RNA. So by this process. So this is all about the RNA polymerase enzymes, in which we have two types: DNA dependent RNA polymerase, RNA dependent RNA polymerase enzyme. Now coming to most important concept, so that is prokaryotic polymerase enzyme. So we will discuss in detail about prokaryotic polymerase enzyme. Prokaryotic polymerase enzyme. So in which we have two types. So there prokaryotic DNA polymerase enzyme, prokaryotic DNA polymerase enzyme, and one more prokaryotic RNA polymerase enzyme. Now first we will discuss about prokaryotic DNA polymerase enzymes. So here we have three types of prokaryotic DNA polymerase enzyme. So first one, DNA polymerase 1, DNA polymerase 1. Second one, DNA polymerase 2. And third one, DNA polymerase 3. Now we will discuss each type in detail, their function. So the DNA polymerase 1 helps remove the RNA primers. Remove the RNA primers. This removal is exonuclease activity. Exonuclease activity means which can remove the nucleotide only from the end of the DNA strands. And once which remove, then it fills the gap. This enzyme was discovered by a scientist called Arthur Korenberg. Discovered by Arthur Korenberg. Hence, This enzyme also known as Korenberg enzyme. Korenberg enzyme. Now coming to DNA polymerase II enzyme function. So this enzyme also involved in DNA repair mechanism. This enzyme involved in DNA repair mechanism. And most important function of DNA polymerase III, this enzyme. This enzyme is responsible for elongation of two strands. They are leading strand, and also called, and one more, lagging strand. Leading and lagging strand. So generally these two kind of strands are found during replication process of DNA. For example, here if you see here, so this is the parent DNA. So this parent DNA, when it undergoes replication process, then there is a formation of replication fork. So for example, here 5' to 3', 3' to 5'. And there is a two opposite strands which is formed along with this separated parent strands. So one strand form continuously from 5' to 3' like this, 5' to 3' towards the replication fork, and one more strand which is formed from discontinuously, from like this, discontinuously. And this discontinuous strand which is formed away from the replication fork, this strand is called lagging strand. And the fragment, the piece of this DNA is called Okazaki fragment. And it is a discontinuous form of DNA strand, discontinuously which is formed. But here in 5' to 3', it is continuously strand will be formed continuously, and this strand is called leading strand. And leading strand is a continuous form of strands towards the replication fork. So like that we have two types of strands formed during DNA replication process. So this leading strand and this lagging strand is only synthesis or elongated only in presence of this DNA polymerase III enzyme. Now coming to next, prokaryotic RNA polymerase enzyme. Second one, prokaryotic RNA polymerase. Yeah, in which we have these enzymes contain two parts. They are protein part and non-protein part. So this protein part is a core part, or it is also called apoenzyme part. This non-protein part is called cofactor. And this prokaryotic RNA polymerase, which mainly helps in RNA synthesis. We know that they play important role in RNA synthesis. See here in this protein part, we have different types of protein parts like 2 alpha and beta prime and epsilon. And here 2 alpha main function is regulative functions during RNA synthesis, regulative function, and these beta and beta prime helps in catalytic function, and this helps in assembly functions. And coming to non-protein part, that is cofactor, in which we have one best example, that is sigma factor. See, this is what sigma factor. So the sigma factor which recognize promoters, recognize the promoters, and it recognizes two types of promoter. In prokaryotic, it recognizes Pribno-Box promoter, and in eukaryotic, it recognizes the Tata box promoter. So this is all about the prokaryotic RNA polymerase enzyme. Now we move on. Important here, one more, that is eukaryotic polymerase enzyme. Yeah, here eukaryotic polymerase enzyme. So here we have two types of eukaryotic polymerase enzyme. They are eukaryotic DNA polymerase enzyme, and eukaryotic RNA polymerase enzyme. Here, coming to eukaryotic DNA polymerase enzyme, so here we have five types of eukaryotic DNA polymerase enzyme. So one is DNA polymerase alpha enzyme, DNA polymerase beta enzyme, third one DNA polymerase gamma enzyme, DNA polymerase epsilon enzyme, and the last one is DNA polymerase delta enzyme. Now coming to their function. DNA polymerase alpha, also called primase enzyme. So these enzymes which helps in primer synthesis. Here our primer are nothing but it is a short sequence of genetic material or a piece of DNA or RNA. It is called primer. So for primer synthesis, there is a need of enzyme called DNA polymerase alpha, or also called primase enzyme. Now coming to beta, DNA polymerase beta enzyme will help in DNA repair in eukaryotic organism. And DNA polymerase gamma, also called helicase enzyme. So these enzymes which mainly helps in replication of organelles DNA, very important. Organelles DNA like mitochondria DNA, mitochondria DNA replication, and chloroplast DNA replication. So this enzyme play important role. Now coming to epsilon, this enzyme helps in synthesis of leading strand. This enzyme helps in synthesis of leading strand of DNA during replication process. And this delta DNA polymerase delta helps in synthesis of lagging strand of DNA. So they also helps in replication process. Or here lagging strands, in which strands there is a small piece or fragments are called Okazaki fragments. So means to combine or to synthesis of Okazaki fragments also mediated by the DNA polymerase delta. So this is all about the eukaryotic DNA polymerase enzyme, in which we have 5 types. Now we will move on eukaryotic RNA polymerase enzyme. Eukaryotic RNA polymerase enzyme. Here we have 3 types. 3 types of eukaryotic RNA polymerase enzyme. They are: first type RNA polymerase 1, RNA polymerase 2, third one RNA polymerase 3. So here their important role we will discuss. RNA polymerase 1 helps in synthesis of rRNA, ribosomal RNA, except synthesis all type of rRNA, ribosomal RNA, but except 5S RNA, 5S rRNA. It means that it synthesis various types of ribosomal RNA like 5.8 S rRNA and 16 S rRNA, ribosomal RNA, 18 S ribosomal RNA, and 23 S ribosomal RNA, and most important 28 S rRNA synthesis. So this RNA polymerase 1 which helps in synthesis of all these RNA but except this 5S rRNA. Now coming to DNA polymerase, this enzymes helps in synthesis of synthesis of HNRNA, heterogeneous nucleus RNA, and it is a precursor of mRNA. So from each HNRNA there is a formation of mRNA, so messenger RNA. So we can remember RNA polymerase 2 helps in mRNA synthesis or also HNRNA synthesis, messenger RNA. Now coming to RNA polymerase 3. So these enzymes helps in synthesis of tRNA, transfer RNA. So not only synthesis of transfer RNA, but also which helps in synthesis of this 5S rRNA. So here 5S rRNA synthesis taking place in presence of RNA polymerase 3. And one more RNA also synthesis here in presence of RNA polymerase 3. So that is snRNA. snRNA. So snRNA, 5S rRNA and tRNA, transfer RNA. So these three types of RNA are synthesized in presence of RNA polymerase 3. So this is all about the eukaryotic RNA polymerase enzymes. So in this video lecture, we will cover entire concept of polymerase enzyme like DNA polymerase enzyme and RNA polymerase enzyme. So also we covered prokaryotic polymerase enzyme and eukaryotic polymerase enzyme. In prokaryotic, we covered DNA polymerase enzyme, RNA polymerase enzyme. In prokaryotic also we covered DNA polymerase enzyme and RNA polymerase enzyme. So this is all about the concept of RNA polymerase enzyme. So all right, so we have completed this entire concept from this topic from molecular biology. Now it's time for self-test. So here I have provided three highly important NCERT based multi-statement MCQs. Now on the screen here, the first MCQs here. Yeah. So consider the following statement regarding the DNA polymerase in prokaryotes. So here in prokaryotes. So here there are 4 statements are given. So which of these statements given above are correct? So analyze the concept what we have discussed just now, and then answer it in comments box. So drop your answer in comment box. And one more important MCQs here. Yeah, this is the second MCQ. Yeah, consider the following statement regarding the eukaryotic DNA polymerase. So here the keyword is eukaryotic. Here asking about eukaryotic DNA polymerase enzymes. So here also there are 4 options are there, are DNA polymerase alpha, DNA polymerase delta, and DNA polymerase epsilon, and DNA polymerase gamma. And which of the statements given above are correct? So one more MCQs. Yeah, here. Yes. Consider the following statement regarding eukaryotic RNA polymerase. So here keyword is eukaryotic. We are asking about eukaryotic and about RNA polymerase. So whether it is RNA or DNA, first we have to identify that, or whether it is asking in eukaryotic or prokaryotic. So like that, we have eukaryotic DNA polymerase here. So here also there are 4 options are given. So which of the statements given above are correct? So this is all about the MCQs from this polymerase enzyme topic. So with this we have complete each and every concept. So thank you and have a nice day.